Okay, let's go to the last talk in this session. It's Jieyue Li from the Carnegie Mellon University, and he's talking about protein subcellular location patterns.
So now I'm going to present my work, which is to use latent descriptive models to classify images by their protein subcellular location patterns. So before I get really into the methods stuff, I will introduce some background about the protein subcellular location patterns. They are just the distributions of proteins in cell components within cell, and they are very important in the systems biology because we can have some information of the status and functioning of the cell from those patterns. Here I show some cell components in a typical animal cell. So there is a rich source of images showing protein subcellular location patterns in the human protein atlas. They are confocal immunofluorescence images. And I show here a typical image from the dataset. Each image contains 4 channels. The green channel is for the protein staining. The red is for the microtubules. The yellow is for the ER, and the blue one is for the nucleus. So each image has been annotated by human for their protein subcellular location patterns. And our work is to build some automated algorithms to recognize those patterns. So the motivation of our work is we can view the subcellular location patterns as the colocalization between protein distributions and the cell components. However, most of the cell components are invisible in the images, so our work is try to infer those cell components in some specific region within cell. So before I get to the models, I will first introduce some image preprocessing we have done for those images. We first randomly select a bunch of regions from cell, and we define that each region has to have some signal of the protein staining, and then for each region we calculate the subcellular location features which have been developed in our group to characterize the distribution of the proteins. So then I will start to introduce our models. The first model we built is called Hidden Logistical Regression, HLR. It originated from the ordinary logistical regression. Here I show a graphical model of the logistical regression. So for each region, we have a class label and a set of features. They have direct dependence in this region, and we can use a log linear model to directly model the probability of this class label conditional on the features and parameters. But in our case, we added another variable which is to represent the cell components in the region, and this variable is latent. We cannot observe the cell components in the image. So we incorporated this latent variable, and mathematically speaking, we just use multinomial weighted parameters in the logistic regression, and we can write out the objective function to learn the parameters. Note that in this model, all the regions from the same cell are assumed to be independently and identically distributed, but this is not enough. Because closer regions in the cell may have higher relationship. So we want to model this kind of dependence between the regions. And it comes to the modified model we call hidden conditional random field, HCRF. In this model, we also show a graphical model here. So for each cell, we can have several regions, and instead of assuming they are independent, we give dependence between the regions, and to be reflected in the mathematical form, we just added another feature, which here we only use the spatial distance between the regions, and then the mathematical model will contain two parts. The first part is the same as the previous, which is a self-associated feature, and the second part is new, which is an interactive feature. So we again use some multinomial weighted parameters, and then we can write out the objective function in the same way to learn the parameters. To learn the parameters, we use the gradient-based methods, and I present here the first derivatives for both of the models. Note that in calculation of the derivatives, we have to know the probabilities of the latent variable given the class label and features, and also the joint probabilities of the latent variable and the class label condition on the features. So we have to do inference in order to learn the parameters. For the first model, HIR, the inference is trivial and straightforward. We can, because the number of the latent variable T and the number of the class labels K are usually both small, so we can expressively enumerate all the combinations of those probabilities. But for the HCRF, it is intractable because we have exponential joint probability table here on the number of the latent variables. To overcome this problem, we will use some approximations in the structure of the model. Our goal is to decrease the tree-wise of the fully connected graph to a smaller number, and to achieve this, we cluster the regions for each cell to some groups, and we decide a small number of the regions in each group. And then we remove the edges between the groups in the cell, but keep the edges within each group. Therefore, we can have a smaller fraction of this exponential part, and make the problem tractable. And as usual, we can give the regularizations for the parameters to account for some overfitting problem. So then I will evaluate our methods on the real data set we just introduced from the HPA images. Before actually I discuss the results, I will recap some basic statistics of the data set we used. There are thousands of proteins and almost 1 million of regions and thousands of features. We use PCA to reduce the features to a reasonable number. And then we apply the two models on this data set. So here I show the results from the computation. There are two figures here. The right side figure shows the accuracies, and the left side figure shows the precision recall curves of these models. For the accuracies, the x-axis of this figure represents the number of the latent variables, or the size of the latent variables we used. And to compare with our models, we use a baseline of using SVM to classify those images to the subcellular location patterns. We can see from the curves, the two curves from our two methods, that both of our methods have statistically significantly better results than the SVM, and the HCIF got the best results. And also, another observation from this figure is that because when t equals to 1, both of the models actually degenerated to the ordinary logistic regression. Therefore, because the accuracies of the t greater than or equal to 2 have better results, we conclude that the incorporation of the latent variables actually does help us to improve the accuracies. And for the precision recall curve, we can see that HCIF did pretty well. It can rapidly go to the accuracy of over 90 or 95 with still a fairly large number of recall. Then I'll present some the classification accuracies for all the classes in the coefficient matrix, because this table is highly imbalancement. So we can expect that the larger class can have higher accuracies, but they still keep fairly good accuracies in all the classes. But then, given the two models, how can we interpret the latent variables? How can we say connect the latent variables to some biological interpretation? So we calculate the joint probabilities of the latent variables and the classes, and we show the joint probabilities in the heat maps from both of the models. The left one is from the HLR model, and the right one is from the HCIF model. From the left one, we can see that the first latent variable actually contains or can be interpreted as those combinations of the patterns which have highly close to the nucleus. And the second latent variable is in an opposite way of showing those patterns. For the HCIF, for the first component, similarly, it contains those patterns that highly close to the nucleus. And for the second latent variable, it contains some cytosolic patterns and has some granular distributions. And for the third latent component, it also contains some cytosolic patterns, but with smooth distribution. Therefore, we can infer some texture-related patterns from HCIF. All of the code and the intermediate results can be downloaded from our website. You can use them if you want. So to conclude that we have built two latent discrete models that can statistically significantly improve the accuracies to recognize those subcellular location patterns in protein images. Finally, I will give our acknowledgments to the HPA group for providing us the high-resolution images, and also we will thank for the funding sources to support our work, and also the ISMB Travel Fellowship to support my presentation here. Thank you very much for your attendance and interest. If you have any questions, you can just ask me.
Lots of time for questions.
For each image, you have 50 regions, right?
Yes. For each cell, we have 50 regions. So the images have been firstly actually segmented into single cells, and we sample regions in each cell.
Okay. So in your classification, do you have different label, different classes for your each regions?
No. We give the same class label for all the regions from the same cell.
Okay. Okay. Thank you.
Thank you.
Any more questions? If that's the case, then thank you very much again.
Thank
and we
you.
close the session now for this morning. Thank you.